This is the Biology EOC review, one of 3 days that are happening. The first one is going to be on April 30th, 2016. This is kind of an experiment. It's just basically me recording stuff on the computer. So people were there, they were asking questions. You might hear some downtime, and I'm probably waiting for responses for people. It's a bit lengthy. It's over an hour long, but I think it does a good job on just kind of giving you some basic concepts, especially when it comes to that EOC test date that's coming up. So watch, enjoy, listen, and let me know if you have any questions. Here we go. So biology, it's been fun, I know, and I'm going to try to summarize everything. I was thinking about just starting at the beginning. So nature of science, how's that feel? Pretty good? Okay, let's see how I do. So, nature of science. Okay. So when it comes down to science, we know there's two kinds of things out there. We know there's science, and we know there's something out there called pseudoscience. Do you guys remember what pseudoscience is? What is it? Yeah, I totally agree. It's a fake science, right? And then real science is a process, right? We know science is basically pretty much going to be like hard facts, that we can measure. It's actually generally, it's the study of the world around us. I probably shouldn't publish this online because my spelling is atrocious. So everybody in the world is going to be like, this guy's a teacher and he doesn't know how to spell world. He had to like add the L in last second. So that's what science basically is, right? Now we said this is a process. Yeah, and we said we have to have like an established system, right? Now we study biology here. And how would you define biology? What do you think, Riley?
Study of life.
Study of life. I like it. I'm going to elaborate a little bit, but it's the study of living things, which I think is pretty nice. Like it does a pretty good job of summing that up, right? We talk about all forms of life, smallest little tiny cells, all the way up to the big grand things, like the whole biosphere, the entire Earth. We talked about ecosystems, body systems. We've talked about, oh my gosh, how energy is made, how it's used, genetics, all kinds of things. So it's a very broad topic. So it's the study of all those living things. And it's done in a systematic way. Now, there's a whole advantage to like using science the way we do, because basically we say everybody in the world does science the same way. There's a method behind this, right? And this brings up the idea of what the scientific method is. Yeah? You guys remember the scientific method? Yes. So let's see. Scientific method. All right, so when we're starting our scientific process, what's the very first thing we have to be able to do?
Make an observation.
I like it. So we're going to start off with an observation. Oh my gosh, my circles are awful. Deal with it. More like a bean shape, but that's okay. So an observation. Now, this can be done just with your eyes, but also we can notice it in another way. We have technology where we can measure things. We could measure wavelength of light. Maybe we detect different kinds of signals. Maybe we notice heat being created, and that helps us see things and notice things, right? Well, once we have that observation, what do we like to develop? Yeah, we like to develop hypotheses. So one of the few things Brandon's ever said that's correct is it's right there. Hypothesis. Okay. Now, I should also mention, but between that time of your observation and your hypothesis, we could always go off and do a bunch of research. Right? because maybe, hopefully, you don't develop a hypothesis just willy-nilly. You're just like, oh, here it is. You always want to base it on something that's actually, that matters. So let's say we're studying a pond, right? And we're like, hey, why are all these fish dying? You're probably going to look at things focused on the lake, and you're going to come up with an answer. Oh, I think maybe there's a bunch of fertilizers or chemicals in the water that are killing the fish. Maybe there's a new animal that's killing the fish. You're not going to come up with something crazy like, because I ate Cocoa Puffs this morning, right? So it's always going to be based in some kind of research, and hopefully you have a background of that. So after we develop a hypothesis, what do we move on and do? We're going to have to test it, right? So the very first thing we do before we test it is we have to design something to test it. So we design an experiment. Okay, so when we design our experiment, there's a few key things we want to keep in mind. And that's going to be the independent variable. I'm going to say it IV. Those are dots, by the way. That's the dependent variable. That's the control group. And then we have control variables. Okay, and we'll explain those again here in a second. But let's go ahead and get through this whole design here. So we've got the design, and then Andrew pointed out that we actually are going to test this experiment. This means actually doing the lab, like, actually doing the experiment. Now, I know it seems a little redundant, and like Andrew, when you gave me your answer, you're telling me what 90% of people would say, and you're right. But I like to add an extra step here because when we design an experiment, we come up with our stipulations. We come up with everything that we want, and then we actually have to test what we design. You'll probably remember, like, earlier in the year, I made you guys design some experiments a few times, and I would like, okay, come up with your independent, your dependent, your control group, and then tomorrow you're going to come in here and you're actually going to do the lab. Well, a lot of the times people would show up and they're like, oh, let's change stuff. So they're not actually testing the thing they designed. So that's kind of a thing to keep in mind. So after we actually test that experiment, what are we going to do? What are we going to gain from doing that experiment?
Data.
Yes, we're going to get data. Or data. It doesn't really matter how you say it. Now we get data in two forms. I'm going to write that over here. Two forms of data. They both start with the letter Q. You guys remember what they are?
Quantitative.
Nice. Quantitative. And qualitative. All right. So quantitative. Do you guys remember what that means? Numerical. It's a number. I'm going to use this symbol right here. That doesn't mean hashtag YOLO. That means number. So it means something that you can actually measure. How tall is something? How wide is something? What's the temperature of something? Anything you can really slap a number on. Qualitative, though, what would you use to say qualitative is? Words. Just yeah. I like it. I'm going to say descriptions, because everything you just said, words, like adjectives to actually describe the thing. You're talking about, actually elaborating about little qualities that you can't measure. You can only feel. So, like, qualitative data could be like, how smooth did it feel? You know what I mean? Like, there's not a number for how smooth a human feels something. And once again, that's kind of subjective. People will feel that differently. There's experiments that are done in psychology, which is a science. There's experiments that are done that routinely ask people how they feel, and that's qualitative data. It doesn't mean it's invalid. It doesn't mean it's worthless. It just means you're kind of taking those people's opinions. Anyway, so we've got our data. What do we do with our data?
We analyze that data.
You analyze that data. That's actually one of those things, one of those key things behind a lab report whenever we made those. And analysis of data is going to be really important because it makes you look at all the numbers and put them into an organized way. We use this in a few ways. We make graphs, we make charts, we make tables, and honestly, like, we have practiced that a good deal. Almost every lab, I make you make a table, I make you make a chart, I make you analyze your data. And then once we analyze our data, what do we end up doing? I like it. Conclusion. Okay, so once we form a conclusion, that's going to be really important because we use this and we base it on our... Good try. Yeah, there we go. We always go back to the hypothesis. It's based on that. Now, the truth is there's two ways this can go, right? Either you can accept or you can fail to accept or reject your hypothesis. So there's two ways this can go. If we, we're going to shorthand it here and say if we reject, which in all technicality isn't true, but don't worry about that. So if we're like, hey, our hypothesis, our guess on what would work, and just help you out, that's our guess. Did we find out our hypothesis was right or wrong? Well, if we were like, no, our hypothesis was wrong, so we reject our hypothesis, we have to go back to the process of actually coming up with a new one. Or maybe we designed it wrong, or maybe we tested it wrong, which honestly is like probably the most common thing. A lot of times you do the lab, you find out, oh, I screwed this whole thing up, let me go back and just try again. And then maybe otherwise. Maybe your data collection was wrong, but either way around, we go back and we redo. That's the goal. Heck, maybe if you can go back and do some more research. But if we accept, we send our information somewhere. We don't just sit on it. Like if you discovered the cure of cancer, you're not just going to like sit on your hands and be like la-di-da, la-di-do, doopy-dop. What are you probably going to end up doing? Probably, yeah. We actually have a special phrase for this, and it's called peer review. Okay? And peer review means you are going to have your peers review it. And your peers are people that study the same stuff you do. They work with you, you know, or maybe they work in another part of the world, but they study the same thing. They're people that know what they're doing. And you're supposed to give them, you know, the opportunity to look at your data, find out, you know, your analysis, your procedure, all that stuff. And hopefully they come back and they say, hey, that's a really good idea. And then, you know, you actually make a discovery there and people kind of freak out. If they come back and they say, hey, I looked at it, I'm another set of eyes, and you need to look at how you collected your data again, or you need to go back and redesign. Okay, now you know I've kind of got something in place, right? How are we feeling about the scientific method there? Pretty good? Thanks for coming out so early, guys. So just a couple like finishing up things about the nature of science. I want to point out to you that there's something called the independent variable. There's the dependent variable. And then we have a control group. And we'll do the control down here. But anyway, so let's say what the definition of an independent variable is. Any ideas? Nice. So factor, and I'm just going to shorthand what you said. We change. Right, this is the thing we are controlling, right? And how many independent variables do we want? We only want one, right? There's only one of these, so that's an important thing to keep in mind. When it comes to the dependent variable, what do you think that is in our little experimental design? Nice. Gosh, Brandon. You're not allowed to be right anymore. Basically, I'm going to kind of summarize what you said, and these are going to be the results we measure. Right? So independent variables cause dependent variables. And it's actually phrased dependent variable because it depends on the independent. I'm also just going to point this out to you, and this is like a thing particular to the EOC. Sometimes they'll use different phrasing, so I don't want you to get all confused or panicky or anything. Sometimes they do actually call these the measured variables, and sometimes they call the independent the tested variables. And that's okay. Like, honestly, when it comes down to it, we just want to make sure we kind of have options available. Then we have a control group. And this is going to be our test group that's going to be missing the what?
Independent variable.
Yes. It's going to be missing the independent variable. And that's important because we want to make sure, hey, Don, just sign in for me right there. That's going to be the variable that we are going to, just remove, and we're still going to allow it to run through the experiment. Now, we don't expect much to happen with this, right? Like, because we didn't change anything. We just hopefully see a result. And we always compare our results to this. Okay, and this lets us know, like, oh, this independent variable really did have a big influence, or it really didn't actually have a big influence. So, and by the way, we have control variables. And control variables are going to be what? Solid response, yes?
Things that stay the same.
Yeah. It should be all the other variables that stay the same. My handwriting is really bad. I get it. I understand. So let's do like a little goofy example here. I'm probably going to ruin everything you thought you knew about flowers, but here we go. Let's say I've got 3 cups. Let's make it 4. Yeah, I'm drawing cups. Those are actually cups, guys. All right, shut up, Brandon. Okay, so we got our 3 cups, and inside of each of those cups, I am going, oh no. Anyway, so inside of those cups, I'm going to put a little tiny seed, right? And in every single cup, I'm going to have the same amount of soil. That's the same kind of soil. There's not going to be like a change in pH. It's going to be all the same stuff. And they're all going to be the exact same kind of seed. And I'm going to expose them all to the same amount of sunlight or, artificial light. Doesn't really matter. And I'm going to change one thing, and I'm going to change the amount of water I add. So in this one, I'm going to have 1 drop of water. And this one, I'm going to have 2 drops of water. And this one, 3 drops of water. And in this one, no water. Okay? Now, over time, what do you expect to happen in this first cup here?
It grows.
You would expect it to grow. A lot, a little, some. What?
A little.
A little? Okay, so let's say like we get like a little growth right there. That's supposed to be a leaf. Deal with it. In this next one, what would you expect to happen?
More good from the first one.
I agree. So maybe just 2 leaves now, right? And then in the third cup, what do we expect? A lot more growth. Heck, we might even develop a little cute flower. Oh my gosh, it's adorable. And then in the last one here, what do we expect to happen? Probably not much. If anything, maybe a little tiny spike of a growth, but it ain't going to grow any leaves. It requires water. We know plants require water. So when we look at this in our little experimental design, okay, and I'm just going to number these for you. In our experimental design here, what is our independent variable?
Amount of water.
Yeah, that's the thing we changed, right? We changed this. So in this case, it's going to be the H2O, right? Okay. And then the dependent variable, what are we looking for? What are we trying to measure?
The height of the plant.
Yeah, we're looking for the growth in general, right? You could measure it in a few ways. You could measure the height. You could measure how many leaves. You could measure if it grows a flower. But whatever it is, you got to make sure you establish it. But we're looking for overall growth. The control group. What's our control group here?
Number 4.
Cup number 4. I like it. Why'd you say that, Brandon?
Because it has no water.
No water. And that's what the independent variable was. So no water means that doesn't have an independent variable, which is exactly what our test group there is. And then we have control variables. Can you guys tell me what some of those control variables are?
Amount of sunlight.
So amount of light,
Kind of plant.
type of plant, anything else?
pH of the water.
Okay, sure, yeah. pH of water. Anything else? Well, I guess probably the temperature outside would be the same. You know, they're going to experience the same temperatures. Probably the size of the cup, because of course, larger the cup, the more soil, maybe a difference in chemicals. So there's a lot of factors there, and you could say a bunch of other things. It's pretty open, but we'd always want to be able to identify exactly what those are, right? So closing that off. Any questions on an experimental design? Are you sure? You positive? Okay. Let's change topics. So that's kind of in general what we talk about. You know, we know what science is, we know what isn't science. And by the way, just remind me, somebody already told me, but like what was an example of a pseudoscience again?
Science.
Yeah, but like an actual real world example.
Psychic or.
Yeah, yeah, like astrology is the one that's like psychics and like, oh, where are the stars? And you know, I've done some math, and all of a sudden you're gonna win the lottery, right? That's astrology. Astronomy is a real science, and they just study stars and planets, and they don't try to make any predictions on your life based on where they are. They just try to go, hey, there's Mars. It's pretty cool. Oh, thanks, cool. So anyway, so let's go ahead and talk about those chemicals that we find in life, okay? And we're going to call that the chemistry of life. The chemistry of life. Straight lines are very difficult for me. So, chemistry of life, one of those first chemicals we talked about pretty extensively was water. It's a big deal. Can you guys tell me a little bit about just what water is? You guys remember anything? You could try making something up, Brandon. Oh my gosh. That's so wrong. It's sometimes right, though. Well, let's just look at what I have here. I wrote down H2O. What does that tell us about water? There we go. 2 hydrogens, and that's 1 oxygen. Now, honestly, on your EOC, they shouldn't ask you any crazy chemistry questions, but there are some principles of chemistry you should be familiar with, especially with water. Water is made up of 3 different atoms, and those 3 atoms are only 2 different things. And that was, as was pointed out to us, we've got one that's here, and that's going to be our oxygen. And we've got one right here, and we've got one right there. And each of those are going to be our hydrogens. Okay? Now, these things together, this shape, does some interesting stuff. I'm just going to overall explain. Hydrogen atoms only have 1 proton and 1 electron. Okay? Oxygens have, I believe it's 8 electrons and 8 protons. But what they do is they form a bond here that kind of steals and shares electrons, which means they kind of act funny. That means overall, these hydrogens have a positive charge because the middle of their design here holds a positive charge, where the electrons, the thing that flies around on the outside right here, that is going to pretty much stay closer to the oxygen. So that means this side is going to stay very negative overall. We call this electronegativity, by the way. Do you guys see how like there's a line here that says positive and negative? That line represents what we call polarity. So water is what we call a polar molecule. Just like, Earth has a north pole and a south pole. You know how a battery has a positive and a negative side. Well, water kind of has a positive and a negative side. And this actually makes them act kind of funny. So when I have a bunch of water molecules next to each other, that means they like to hang out in particular directions, which is good. So the positive side always touches the negative side of another water molecule. Now, this whole thing is pretty amazing. And when we see the bonds that form between these water molecules, we call this a special kind of bond called a hydrogen bond. Now, the hydrogen bond is wildly, amazingly important to all life, all living things, because we know water is one of those very important molecules for all living things, no matter what. So basically, the hydrogen bonds are just those little attractions between each other. And hydrogen bonds are responsible for all of the special properties of water. And we said water has like a good number of special properties. Now, can anybody remember at least one of them?
Adhesion and cohesion.
Oh boy, adhesion. And you said cohesion. Okay, so let's go ahead and define what is adhesion.
It's its ability to bond with itself and try to stay together with itself.
Close. You're actually defining cohesion. So this is when water bonds to other water molecules. Okay? So water loves itself. It is so selfish. But it kind of makes sense because I just showed you, like down here in the drawing on the left, those hydrogen bonds, they form between other water molecules like crazy. Like it's all the time. Think about it. Like you notice this in nature because puddles exist and water droplets exist. Like a water molecule is very tiny, extremely small, yet they always seem to find each other. Have you ever been, like, I don't know, to Denny's? Or, okay, any restaurant will work. Okay, you know how they have their tables and they give you your water or your soda or whatever. And we live in Florida, so it's always hot. And you always get the condensation that forms on the outside of your glass, right? When that falls on the table, you can play around with that water and you can just drag your finger through it and you drag a bunch of water with you and make a little trail because the water attracts to other water molecules. This is one of the reasons why plants are so darn effective at keeping water in there. They pull water up from the ground and more water follows it because it sticks to itself. Now, adhesion is going to be a bit different, kind of the same side, different side of the same coin. What do you think adhesion is?
Its ability to bond with other molecules as well.
There we go. Yes, that's when water bonds to other molecules that aren't water, right? Which is pretty remarkable stuff. This is, once again, it plays a big role in how plants work because if you think about it, the plant or a tree isn't made of just water. It's made of other stuff. You guys know paper towels. Hopefully you use them pretty regularly. That holds water like crazy. You know what I mean? If you wear like, your t-shirt, you know what I mean? Like it's hot outside, you're probably going to sweat. Sweat is mostly water. It's going to stick to your t-shirt, right? Because it wants to. Water loves to stick to those things. And then we have some other cool properties. Anybody remember any of those?
High specific heat.
High specific heat. That's a tricky one too. High specific heat. This basically means water needs to absorb a lot of energy to change temperatures. Now, high specific heat is amazing because it basically says my body's mostly water, your body's mostly water. Whenever I stick my hand inside of my freezer at my house, or I go outside and it's below freezing outside, it doesn't just suddenly turn into ice. That would be bad for life if you are suddenly a block of ice. So high specific heat makes sure that you can usually maintain water's temperatures, usually liquid, which is a good thing. We want that to be true. Another thing that we find very important for the properties of water is going to be it expands when freezing. And this is actually kind of cool because when we have our water molecules, what they do is they evenly space out. This is when it's ice, right? But when it's just like a liquid or a gas, it can space out in crazy ways. Like it seems random and they all move around, but ice doesn't move around. And so what happens is they evenly space out. Like this is why, oh my gosh, like this is how the Egyptians like broke giant rocks to like, you know, speed up the making of buildings, like the pyramids and stuff. They would make a little crack in the rock. They'd pour water in there, liquid water, and then at night it would freeze because the desert does that. It gets very cold, and that ice would expand. And the cool thing about water is it's what we call a not non-Newtonian. Yeah, actually it is. It's non-Newtonian fluid, which means we can't compress it. We can't. This is how like you know potholes. Like I kind of already explained this before, but like Dairy Queen. You know Dairy Queen here? Like the Dairy Queen has the worst potholes in front of it ever. It really is bad. They just recently patched it up. And by the end of the summer, I guarantee there will be potholes because every time you have like a little tiny divot and water gets in there and you drive a car on top of it, you're forcing the water down. It's not going to compress and it's going to force that rock to move more than the water. So you're going to have like the rock get chewed up and move around. So it expands when freezing, and that's kind of the reason why. I think there's one more property of water. Do you guys remember what that is? No. Now I'm trying to remember. Oh boy. Expands when freezing. High specific heat. Cohesion, adhesion. Oh, yeah. It has neutral pH. So, do you guys remember what the pH value for water is? Yeah, it should be right at 7. Like pure water is going to be 7. I think I'm pretty sure like ocean water is slightly basic. Pretty sure. Might be acidic. No, it's probably acidic. And then we have like everything you consume, all the water that you drink. There's going to be varying amounts. Like tap water might be slightly acidic or slightly basic. But mostly water is just a pure 7. And actually, when we think about that scale for water, right, the scale goes from 0 to 14. So right there in the middle is the 7. That's where water should be. Anything that's 7 or higher, we call that a base. And anything 7 or lower, we call an acid. So those things are, pretty much every chemical can be measured for a pH. And it's important we recognize that because we don't like to mix acids and bases all too often. That goes boom. Have you ever mixed vinegar and, you know, Alka-Seltzer or baking soda? Hopefully you have. If not, you can go home and do it, make a mess in your sink. Or maybe dislodge something stuck in your drain. That's basically what drain cleaner is. You force an acid-base reaction and it helps break that stuff up. Anyway, if we get really extreme on the zeros or really extreme on the 14, we can start to eat away and break down a lot of materials, which is not great for life, but we typically avoid it. Most things operate here in that middle range, right around 7. So most things live in that range. So how are we doing on water? Yeah? All right. So since we're still talking about the chemistry of life, let's talk about some of those actual major things that we like to call macromolecules. You guys remember the macromolecules? Let's see if we do. So we've got four main categories of those. We've got protein, we've got lipids, we've got the fun one, carbohydrates, and the last one is going to be kind of a weird one, but they're called nucleic acids. Okay? So these are those four main, really bad straight lines, four main molecules that we find in pretty much all biological things ever. Now, they each have their own functions, they have their own purposes, and they're basically assembled from some of the same building blocks. And just to kind of point out to you, the four main elements that we find involved are going to be carbon, hydrogen, that would be oxygen, and nitrogen. Carbon, hydrogen, oxygen, and nitrogen. CHON, C-H-O-N. Those four pieces pretty much make up a majority of these macromolecules. Sometimes we change that up when it comes to the nucleic acids. Sometimes we involve phosphates. And sometimes we can see some weird stuff show up. We can see like sulfur show up and some sodium and stuff like that. But they're usually in small quantities. So let's define something here. These are all called macromolecules. So that means they're usually very large and complex molecules. That means they're made up of very small things, small subunits, which means they're made up of these things called monomers. So just down here, I'm going to remind you, a monomer. A monomer is any small piece that builds up to a larger thing. Now, that means, like, let's say I've got like I don't know, like a chain necklace or something, right? This whole thing together might be a train or a chain, right? But each one of these is actually going to be, this is the worst arrow ever, is actually going to be a single monomer, okay? So I want to know, what are the monomers that make up a protein? Amino acids. Keep in mind, this is all going to be monomers, right? So amino acids. Do you guys remember what they are for lipids? I'll take that, actually. The truth is, in the technical terms, lipids don't actually have a separate monomer. They're actually made up of two separate things. So that's going to be usually like the fatty acids, which are at the tail, and usually there's a head associated with it too. But we'll talk about that here in a minute. And then we've got carbohydrates. You guys remember what they're made up of? It's a funny word. I'll give you a hint. It's got the word mono in it. Yeah. Now I'm probably going to misspell this, so don't judge me. I think it's S-A-C-C-H-R-I-D-E. I don't know. I don't care. Who? Nobody would ever know. You know, like, it's lost to the, histories of time. Nobody remembers. Dictionaries are lies. So, nucleic acids. Do you guys remember the monomers that make up nucleic acids? We studied these extensively, and there's like 4 or 5 variations of them. Yay, it's nucleotides. You're right. So we've got nucleotides. They're pretty darn important. So let's just briefly talk about like proteins. Well, yeah, let's do it here. So we've got proteins, right? Amino acids are going to link together. They're going to form together. Now, I just need to tell you, like, proteins are like one of the most important molecules in your body. I'll say it. They're the most important molecule in your body, maybe other than water. But without proteins, water wouldn't have much of a purpose. So proteins, wildly important, and amino acids are going to be linking together to form them up. Now, proteins are found pretty much to do everything. They literally take place or are used in all bodily functions. Okay? All bodily functions. I mean, it's just, you've got to use proteins. You use proteins to grow. Your muscles are made up of mostly proteins. You've got a lot of brain material that's mostly proteins. Like, they're like the little workhorse of your body. Like, if there's a process, like a little activity that happens, proteins are causing it to happen. I should also point out to you, there's a variation of a protein called an enzyme. And we're going to talk about that here in a little bit. So we've got lipids. Do you guys remember what lipids kind of are used for? They store energy. Yeah. Usually it's over the long term. Now, you store fat on your body. And by the way, that's what lipids are. You know them as fats. They store energy, and we keep them hopefully for a long time. Now, you've probably heard about people that like work out and they try to lose fat. You know, they try to look cut and ripped and blah, blah, blah. The truth is they still have fat in their body. You need it. It's also really important in cell membranes. They're probably the most important structure in a cell membrane, other than proteins, which, you know, are, of course. And really, they kind of take up some structures in the cell too. That's an S. So we've got carbohydrates. They're made up of monosaccharides. Monosaccharides. There's also another form of these things called disaccharides. And their big version, which really goes to carbohydrates, are called polysaccharides. Yeah. Do you guys know what carbohydrates are mostly used for?
Short-term energy.
Yep. Short term. This is exactly why you always get more, like, you literally feel like, hey, I'm more awake. I've got more energy after you eat food. I'm not saying like 30 minutes later if you might get tired. I'm saying like as you're eating, even if you're dog tired, you feel like you got more energy because we're burning those sugars inside of us, which, by the way, you know these as sugars. There's also a few other versions. They're called starch. You've probably heard of things like, I don't know, pasta, breads. They're full of starches. They're full of sugars. And they give you like that shorter term energy. Starches are a little bit more long-term energy. They can get stored in some muscles, and we can use this as part of glycogen, which is actually stored in your liver. But anyway, we use that for our short-term energy. This is why, like, little kids, if you give them a pixie stick, they just go nuts because lots of sugar means lots of energy, and your body says, oh, I got all this free energy, I better use it. So, like, your heart rate increases and people get kind of hyper. Though it's not always totally true. Anyway, so mostly it's just used for energy structures, but energy uses, but some cell structures, sometimes it's used for that. And then we've got nucleotides. Do you know what nucleotides are used for? That's scary. Genetics. They are your genetic information, right? And we said there's these nucleotides, and I'm just going to go ahead and remind you of what they are. We've got G, C, A, and T, and sometimes a U. You probably remember that guanine, cytosine, adenine, thymine, and uracil. And those are going to be those 5 nucleotides that we find in nature. I would, you know, we'll kind of review there, but if you don't show back up in the future, we're not going to get to genetics today, of course, but you should know like they're pretty darn important. Uh, and just to point this out to you, like it's a very complicated replication process. So these aren't just like willy-nilly. Now they're vitally important. Like this is literally the blueprint for your body. It tells you how to make your carbohydrates, your lipids, and your proteins the way you're supposed to. And we got to have them in place. And some of the very first molecules formed in terms of life were probably nucleic acids, which is pretty remarkable. So I should also point out to you, one of these chemicals, one of these elements that they all have in common are carbon. That's one thing that every single one of these has. And carbon is represented with the letter C. And carbon is very important because it can make bonds with other elements, other atoms. Do you remember how many bonds it can make? 4. And we can actually draw this by doing it like this. Yeah. And carbon likes to bond with itself. It's always willing to make 4 bonds, covalent bonds. And that makes it like the basic building block of everything. Carbon is everywhere. Like, and carbon's like super cheap too. If you've ever seen dirt, a lot of it is just carbon. Actually, that's what gives it its like earthy color, like brownish, blackish. If you find pure carbon, it is black. Like carbon fiber, that stuff's like crazy expensive and like works in cars really well. Like they can build not frames out of it. Well, sometimes frames, but like hoods and like actually the car body. It's very tough and it's carbon, and they just like arrange it in a particular way. So if we can play around with carbon, we can literally make a lot of building blocks of life. So how we doing there? Any questions? Concerns? Okay, how about we take a 5-minute break? Sound good? Okay, so once again, we left off talking about those macromolecules, and we mentioned there are things out there called enzymes. Now, enzymes are kind of weird. It's very strange. These are going to be the molecules that act as a catalyst in our bodies. And a catalyst is anything that speeds up a chemical reaction by lowering the activation energy. Now, I know that sounds really crazy, but I'm going to write it down. Speed up chemical reactions by lowering activation energy. Okay, so it speeds up chemical reactions by lowering activation energy. So let's look at a graph to actually make sense of what that actually means, because it's kind of crazy. So let's say I got a graph here. Okay, typically what you have is you have a level of energy, and we have to add energy to something to actually have a chemical change so we can create what we're looking for, right? So let's say I got a molecule that I want to break up. So I've got like, here's my 2 molecules, right? And they're bonded together. Over here, I'm finally adding in the energy to like get them all excited. That's energy, by the way. I know that seems crazy. And then what happens down here is I finally release that energy and I break the bond. And I have my 2 little pieces that can float off and they can do whatever I want them to do. Like digesting stuff, right? If you eat an apple, I want to be able to break down the sugars inside of the apple so I can use it for energy. So we have to use so much energy. That's what our level there is. This is called, let me redraw this a little bit better. I don't mean straighter, I just mean better. Right, so this is called our activation energy right here. Okay, now if I change it up, we can have enzyme used, which means we start off in the same place. and we end up using less energy, and we still up in the same place. So when we save this, this is going to be from the use of an enzyme, right? And that enzyme is going to be super duper important because they're designed to work in all kinds of cases. As a matter of fact, we use something called the lock and key model to describe how these enzymes work. Now, a lot of the times, an enzyme is just a protein that has a special job. And a lot of times that protein looks kind of like, how do you draw it? Oh, okay, let's try this. Think like puzzle pieces. I know, that looks weird. Deal with it. Shut up, Brandon. Shut up. And so this is going to be our enzyme. Okay? And then let's say we got this thing here. We've got a monomer. Oh my gosh. Monomers, we know what those are. Those are building blocks of cells. So, of macromolecules, that is. Let's say we want these things to fit together, right? And these little pieces that we work with, they're called substrates. Okay? And basically, the enzyme is designed to fit those actual substrates on there. If we had other pieces out here that look kind of like this, they're not going to fit, right? Like they're literally not going to fit on there. They don't have the right ends. So we need a bunch of different enzymes in our body to help us out. Like you have an enzyme in your saliva called amylase. Have you ever taken like a cracker or like a cookie or like cotton candy and put it in your mouth and it just seems to melt? That's amylase doing the work. It's breaking down that sugar. We have tons of enzymes in our stomachs that help us break down things. And this makes it a lot easier because I don't want to have to use a bunch of energy all the time to just digest food. You know, like think about a snake. You know, they eat their food, it sits in there and it stays in their digestive tract, and it takes them a long time to digest it. That's why they get that lump in them and it stays there seemingly forever. We don't want that. We'd like to break down our food faster, be more efficient, and we're like high energy organisms. Like snakes are pretty chill, literally. They're cold. So they don't have to like break it down as quick. So the enzymes kind of work in that way. And I'm just telling you now, like this definition will probably end up showing up in some way on the EOC. More than likely. They always manage to ask a question about enzymes. And all you got to know, it's also a kind of protein. But that doesn't mean that every catalyst in the world is a protein. Actually, enzymes are biological catalysts. All right, so there you go. That's enzymes in a nutshell. Any questions there? No? We're sure? Okay. So, I used to be able to erase stuff. Look at that. I've got an erase all ink on slides button. Isn't that? Oh, that's so good. Anyway, so let's go ahead and talk about the things that those macromolecules actually make up. They're called cells. Yeah? You remember cells? Let's see. Riley looks uncertain.
I remember cells.
Okay. We said cells are defined as the smallest living thing. And actually, we can say that they are the, how could we define that? We define them as the smallest basic unit of life. Okay? Smallest basic unit of life. Can you guys see this? You'd let me write that way and you wouldn't even say anything? You guys are bums. Total bums. Smallest basic unit of life. So let's go ahead and say we've got two major kinds of cells. We've got prokaryotes and we've got eukaryotes. So prokaryotes, eukaryotes. What makes them different? Nothing. Which one did you say? Eukaryotes. These have a nucleus. Okay, that's true. Eukaryotes, these are the cells that you're made up of, right? You carry these around. And they have a nucleus, which is going to be a very important feature, which means prokaryotes must not. They do not. No nucleus. And we'll kind of define what some of these organelles are. It's going to be pretty extensive, but we will define what they are just here in a little while. So we've got no nucleus in a eukaryote. No nucleus in a prokaryote. We have one in a eukaryote. What else do we know? What do prokaryotes really have?
Prokaryotes are single-celled, aren't
They usually are. Usually single-celled. That is true. What does that tell us about eukaryotes? Yeah, or at least they're capable of being multicell. So we'll say single sometimes, but multicell. Okay? Which means eukaryotes automatically now can become more complex than your typical prokaryote, right? What else do we know that's different between the two? Usually they do, but that's another kind of difference between cells that I'll be looking for later. Good try, though.
Prokaryotes don't have cell membrane.
They do, actually. They both actually do. We could actually make a column here that says all cells. And we know all cells have a cell membrane. We also know that all cells have something inside of them. That's true for everybody. Do you remember what that is? Yeah, that's true. But I was thinking more of like genetic information, like DNA or RNA. They all got to have that. Even if they don't have a nucleus, they still got to have it. And I should also mention they have ribosomes. Because they all have to be able to produce the same thing there. Eukaryotes are also going to be really important because they are, well, let me try this. Yeah, we'll just phrase it like this. These have membrane-bound organelles. Right? So membrane-bound organelles, which means they have all those little tiny body parts inside of a cell. Whereas prokaryotes, no organelles. Okay, and that's probably the most distinguishing feature. Overall, that makes prokaryotes kind of dumb. You know, they can't really follow through with a lot of complex tasks. Eukaryotes, very complex. Think about it. Like, your body has lots of different kinds of cells. And you don't just have one generic cell. You don't have like, oh, I have a cell. You have like brain cells and cells used for muscles and cells used for like your skin and your epithelial cells. You got like cells for your intestines. I mean, they're all different and they all do different jobs, right? So that means they're very specialized. So simple and special. All right, those are kind of just the basic phrases to describe eukaryotes and prokaryotes. All right, and that no nucleus nucleus thing also very important. If I were to draw you a prokaryote, they'd probably look like this. Oh my gosh, here we go. Best drawing of the year. Are you ready? Oh my gosh, it's gonna be so good, so good. Huh. Yeah, I know you're jealous. That's okay. Oh, so good. Okay, so this work of art is going to be a prokaryotic cell. Inside of here, what do you think that is? Yeah, that's their DNA. We'd actually call it chromatin. Just a fun little fact. Um, and it's loose. Is it contained in anything? No, there is no nucleus, right? So it's loose, it's just floating around. You might get lucky and find some little ribosomes floating around because we said ribosomes were part of it. You'll still find that cell membrane on the outside. And sometimes you'll see these little legs or arms on the outside called flagella, right? And they're going to be very important because that's actually how they move around. And seriously, like prokaryotes, they just are just trying to eat and survive. That's it. Sometimes they try to reproduce. So they bump into things, go, can I eat this? Or can I mate with it? No? Okay, I'm gonna move on. But eukaryotes, oh my goodness. Eukaryotes have all kinds of stuff inside. So we've got a nucleus, we've got the DNA inside of there, we've got like this branching thing off to the side, looks kind of like that. There's another thing that branches off of that. We've got some other organelles inside of there. Oh my gosh, so many. Sometimes you get some other ones that kind of look like this. I know the drawing isn't great, deal with it. We've got another kind of foldy thing, looks like this. And we've got little bubbles floating around. I mean, it's just a crazy little party inside of your cells. Oh yeah, and there's like little dots out here too. So eukaryotic cells, way more complex than just your simple prokaryotic cell. And yeah, they're single celled, so you're only going to find one of them. There are weird exceptions where they can be multicellular, but they're still overall pretty dumb. So we know the general ideas between cells, right? Yeah? Okay, I kind of screwed up earlier when I was recording this, so I'm doing this when I wasn't doing the lecture. So it was probably the most important slide with all the different organelles. So I'm just going to quickly write them down for you. Some of the organelles that we find inside of cells, of course, are going to be the nucleus. And its job is to basically hold genetic information. And then inside of the nucleus, we have something called the nucleolus. And it's all probably misspelled, so, sorry. The nucleolus, it really is very dense with DNA, but also it's really responsible for making ribosomes. And that leads us to our next organelle, the ribosome. It's actually one of the few not membrane-bound organelles, so it's in all of your cells, but the ribosome's job is to make protein. And those proteins, of course, play a huge role in all of your cells' processes. And by the way, whenever I think about like how cells are organized, I always like to imagine the story of protein. And the story of protein is basically going to be, basically it's going to be how they're made and where they go and what organelles are used to make them, or at least to not just make them, but move them around. So the ribosome is going to be contained after it moves out of the nucleus. It's going to be contained on the outside of something we call the rough ER, which stands for rough endoplasmic reticulum. And it holds the ribosomes. And they make their proteins, of course, and they drop them inside of the rough ER. And then once it drops off, it transports it to the smooth ER. I'm doing this a lot faster than I did previously, but the smooth ER will basically transport a lot of those proteins around, kind of separates them, And then we usually move out of the smooth ER to an area just like in the actual cell. We have to use a special organelle for that, and that's called a vesicle. And a vesicle is like a vehicle. It's the vehicles of your cells, and they just transport not just proteins, but all kinds of things around the cell. And they're made up of the same thing as your cell membrane, which is pretty unique, pretty important. And they transport usually to an area called the Golgi complex or the Golgi apparatus. And its job is to organize and deliver protein. Okay? And so once it kind of does that, it's going to travel back through vesicles and hopefully it'll go out through the cell. But usually when it goes out through the cell, it passes through the cell membrane. And on the next slide, I actually go over areas where we see the cell membrane and areas where we don't see the cell membrane playing a role, especially on the outside. But there's also another thing called the cell wall, which is true only in plants. And we have the cell membrane. Its job is basically it regulates what moves in and out of the cell. The cell wall usually provides structure. And support for the cell. And then we have some things like lysosomes, and they break down parts of the cell, kind of like the recycling center. Like leftover parts, old parts that need to get recycled and reused, hopefully. And then we have a vacuole. Vacuole. Oh boy, I misspelled that terribly, but that's okay. Vacuole. And this is where we store things in cells. Water is usually stored in plant cells. Animal cells usually just like keep byproducts like garbage, stuff that's not necessary for cells. They store them in vacuoles there. And then we have, oh boy, we have the mitochondria. And we have the chloroplast. And mitochondria, they're used for something called cell respiration, which is basically how you make energy from sugars. Both plants and animals have them. And then we have chloroplast. This is used for photosynthesis. This is where that happens. Now, only plants have chloroplast, but they also have mitochondria. Animal cells do not have chloroplasts. And then we have stuff called cytoplasm, which isn't really an organelle, but it's the jelly-like substance inside of the cell. And then we have the cytoskeleton, which is the kind of the proteins that maintain shape of the cell. And I'm not sure if there's anything else that really stands out as super important, but you will be able to distinguish on the next slide what's going on there. I'd recommend to help you review. Like I could like previously I drew the cell. I would not recommend looking at my drawings. I'd tell you to look at a drawing or a diagram from a textbook or from a resource guide or online, and you're going to be able to find all these parts. And really, that story of protein is what you want to keep in mind. So we always move from nucleus to the ribosome because that's where the ribosomes are made. The ribosome makes proteins, and that's found on the rough ER, and then we move to the smooth ER, and then hopefully through vesicles we move to the Golgi apparatus. And then from there, maybe we move back through vesicles, and we can kind of do things that way. I should point out to you that from this review session, we didn't really mention a whole lot from the cell membrane. And the cell membrane uses something called the fluid mosaic model. And we talked about how there would be a lot of phospholipids, which are some of those lipid structures that we talked about previously. And then there's some proteins that are wedged in there. Actually, it's a lot of proteins. It's actually a pretty recent discovery. There's a lot more proteins in our cell membranes than we thought. But anyway, it's a mosaic because it's got a lot of different looking parts involved with it. And the proteins are used for a lot of different things. In this example here, I made a channel protein, which moves things in and out of cells. The phospholipids allow it to be kind of fluid, allow it to move around. We even have like big chunks of things stuck in there called cholesterol a lot of the time. And they make sure it's not perfectly spaced out, so it's mobile. And this is the good version of cholesterol, not the bad version. But anyway, the fluid mosaic model, pretty big deal. And we will go more into the mitochondria and the chloroplast in separate slides because that's once again like really involved with cellular energy. So that's kind of the overview of the organelles we find inside of cells. Okay, so once again, sorry, we got plants, we've got animals. We know they're both eukaryotic cells. And we said plants have a cell wall. And we know that they have chloroplasts. Now, chloroplasts are going to be basically one of the energy cells that plants have. And their job is they perform photosynthesis, right? Now, I know we haven't talked too much about what photosynthesis is, but we know basically plants absorb sunlight and they're able to turn that sunlight and like some other chemicals into sugar. And then they can later break down that sugar and actually get energy from it. So there's our chloroplasts. Now we've got another kind of structure inside of plants, which is kind of weird. We didn't mention it on the last one, but they have vacuoles. No. Vacuoles that are large and centralized. Plant cells, generally speaking, they look kind of like this. They're more rigid. They have like corners to them a lot of times. A very thick cell wall, right? They have their nucleus and all their stuff inside. They have the endoplasmic reticulum and all that stuff. They have their Golgi apparatus, and they have this huge area inside that's just the vacuole. And this is used for water storage because plants need water. So you know how we were talking about celery? If celery like kind of dries out, like you keep it in your fridge for too long, it will wilt. You know, it'll actually get like soft and kind of spongy. It wouldn't be very fun to eat. But if you took that celery, believe it or not, and you stuck it in a vase of water, it would start to fill its vacuoles back up, and it would actually like stand upright again. It wouldn't like wilt as much. That's why like flowers, if you put them in a vase, you got to keep them in the water. If you run out of the water, they'll get all sad. Of course, they're going to die at some point, but you can kind of keep them going by keeping those vacuoles full of water. So they're kind of important in that regard. Animals, though, what's something that only animals have? What do you think, Riley?
Mitochondria.
Yes, they only have mitochondria. Whereas plants, they have chloroplasts, but they also have mitochondria. I mean, that's kind of weird. Most people actually don't realize that, and that might be a question that pops up on the EOC. Plants don't just have chloroplasts. They also have mitochondria. They also have to break down their sugars, and that's what the mitochondria do. They break down sugars to give you energy. So we only have mitochondria in our cells. What else do we know is very special about our cells? No cell wall. That would be kind of really terrible if I had a cell wall on all my cells because then I would be rigid all the time. I couldn't move my arms. You know, trees can't decide to move their arms because first off, they don't have arms, but second, like the cells are too stiff. They couldn't actually move them. They'd break. That'd be terrible. And then I'm just going to mention this one to you. They have very small vacuoles. And that's because usually we have a lot more complex processes happening inside of us, and that makes our job a bit more complicated. So we have a lot of byproducts that we need to get rid of. So we're going to have these small vacuoles, and they don't really store water for us. They just store like garbage. They're like our landfills. They literally build up all the junk that we don't need. So this kind of leads us to one of those ideas, really our first main scientific idea, called cell theory. And this is a bit of a history lesson, and I'm telling you now, like on the EOC, they might ask you about this in one context. And there was this dude, his last name was Hook. Yeah? You guys remember this guy's name? And Hook is the guy who's credited for discovering cells. Actually, he coined the term cells. And basically what he was saying is that there's little tiny rooms, is what he saw. So he looked at cork cells. And you know, cork is just the bark of a cork tree. And what he saw on the inside were these little boxes, little tiny empty boxes. There was nothing in there because it was dried out. And cork's the same stuff that people put in like the stopper of a bottle, like, you know, a wine bottle. It's really light because it's mostly air. And it's dried out, and that air causes it to float, and it stops things from climbing into stuff. And he saw those little tiny things. Now, when he did this, he had some very primitive technology, but he still named them cells. And then later on, some other people came along. I think his name was Virchow. I think Virchow came along, and he was the dude that saw the first living cells. Right? Nope. I screwed that up. I knew I screwed that up. It wasn't Virchow. It was Leeuwenhoek. I'm going to try to spell his name. Leeuwen. Hoek. There we go. He saw the first living cells. He saw, he actually called them Animalcules, I believe. And he basically looked at pond water. Now, he got kind of weird. He not only looked at pond water, he also looked at poop under a microscope. And he was able to see things moving around. He's like, hey, this stuff is alive. It's not just empty boxes. These things are alive. And that led to a lot of people going like, well, I want to see. And then we had some other dudes come along, and they kind of helped us out by discovering, oh, I don't like it. Let's try this. I'm checking through my notes. Give me a second. Give me a second. All right, well, that's fine. Anyway, there was a few people's names. I remember, like, explicitly for the EOC, you're not going to have to remember their particular names, but I mentioned one of them already. There's a guy named Virchow. And then there was, oh my gosh, Schleiden and Schwann. I'm going to try to spell this. Anyway, you don't have to know their names, but I do want you to know they came up with what was basically called cell theory. And there are three postulates, or three parts to it. Number one is what? Okay, so the cell is the basic unit of life, right? Okay. Number two is going to be what?
Cells come from other cells.
I love it. add living, because you're not born from dead cells, right? That'd be kind of weird. And the third one is going to be all living things are made of cells. So those are the three postulates. You will probably be asked a question about those three things. So be wary, okay? I also want to point out to you that this kind of brings up the idea between a difference between a theory and a law. And this is like a common misconception kids usually have. Let me check my notes again, because I want to make sure I'm defining it as best as possible for you. No, 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 no. Anyway, we'll just say it this way. We've got a theory and we've got a law. Okay? I don't want you to ever think a theory is a bad idea. I don't want you to ever think it's a weak idea. A theory is literally the best thought humans have ever thought of. This kind of goes back to that scientific method we already talked about. We said, you come up with an idea, you test it, and you kind of go through that process of peer review. If people are like, yay, you found something, people are really happy, and if people keep supporting your idea, testing it over and over and over and over, you're probably not wrong on something, and we get the credit of calling it a theory. So this is an idea that's been highly tested and not disproven. And I should also point out, these can change. Okay, so let's look at cell theory. We know all living things are made of cells. Like, we have studied far and wide all kinds of stuff that's alive, and we always seem to notice they're made of cells every single time. That's never been disputed. We don't think of something that's not made of a cell that's not alive. The only thing that's kind of iffy right now are viruses. We're kind of like, they're not really alive because it's not technically a cell, but whatever, okay? So, like, nobody in this room would question that we're made of cells, right? Like, there's no doubt about that. There's theories on how motion works, right? We actually have some other things established for that, but there's theories on how things work that way. There's the theory of evolution. Genetics is a theory. So I don't want you to think like some people dispute these things. When it's a theory, it's been highly supported and never disproven, but they can change, like the theory of cells. We've come across this and we've said, oh, notice this little piece does this job. We didn't know what it did before. That changes our idea of exactly what a cell does and operates. So it can change, okay? Laws, on the other hand, these are what we consider scientific facts under certain conditions. Okay, so like the laws of motion, right? Newton's laws of motion. They are always true. They're just always the reality under certain conditions. Now, we can change those rules, but there's some weird stuff going on. So whenever we're seeing things happen, and we can always predict things based on it, we call that a law. So you don't go from a theory to a law. You never graduate. They're separate thoughts. Most of the time, laws are mathematically shown. So in biology, we don't have a bunch of laws. We have a lot of theories because we test ideas, we keep supporting them, and we build off of them. A lot of the times, theories give way to other theories, and sometimes laws give way to other laws. But they don't like graduate. They don't become one or the other. And I don't want you to think one is better than the other because they're both great. Okay? Sound good? All right. So I think we've covered tons and tons of stuff. I just want to mention one more thing, and then you guys can go. So we've talked about a lot of things that happen, right? We've talked about cells. We've talked a little bit about their energy. We've talked about the parts that make up cells and how they all kind of work together. But there's one key concept that's true in all of biology. A living thing maintains itself. Systems try to maintain themselves. Okay? And what we use in our body is called homeostasis. So if I make another graph here, right? Throughout the day, we can talk about our body temperature, right? Now, you know overall, like the body temperature is supposed to be 98.6 degrees Fahrenheit. And that changes, by the way, periodically. Like sometimes they change the rules. Heck, I might even be wrong here. I know at one point it was 98.6. It used to be 98.7. But we'll notice like throughout the day, your temperature can go up and it can go down, it can go up and it can go down and go up and go down. And usually like when you're tired, your body temperature is going to go down. This is why we wear like blankets when we sleep. I'm not saying you always use one, but usually you wake up colder than when you went to bed. And that's what the blanket does. It helps retain that body temperature. So your goal is to maintain this middle level and maintain that homeostasis because you want to conserve your body's temperature as best you can, close to that nominal area. When we talked about enzymes, we talked about how the enzymes make it, have to use less energy, right? Well, if we want to use less energy, that's going to be important because it's going to kind of fluctuate over time. We've talked about ecosystems, and we said, if there's too much of something, it'll bounce back out. It'll go up, it'll go down, and it always changes. It's a very dynamic system, okay? And we always try to maintain homeostasis because it means it can still change periodically, but it tries to hold one level, okay? So, that's it. That's all I got for today then.